Hi everyone, I'm Stephen Cook from the Emmons Lab, and if you are watching this video, it means that you're interested in annotating some neurons in the C. elegans ventral cord using CATMAID. So the idea of this project is that we may be possible to annotate a nervous system in a crowdsourcing fashion similar to something you may have seen on Galaxy Zoo. So by this point you should have emailed Stephen Larson and received some login credentials for our CATMAID instance running on Amazon AWS. So first you're going to enter in your account name, password, click login. When you're here, you're going to see some projects on the left. So Stephen will have told you in the email which project you'll be working on. Today we're going to work on test 3. So you'll be presented with an image such as this one, and you'll have a group of 2 to 5 images of which you'll have the ownership over to begin to annotate the neurons and the synapses. So there are 205 images in this data set, and as you can imagine, to do that all yourself may take an appreciable amount of time. So just starting with a small number of images will make it more manageable and allow us to work on this project in parallel with one another. So once everyone's done, I'll be able to review these data. So these images are from the White et al. 1986 paper, also known as Mind of the Worm. I have also annotated these images, so I have an idea of what should be correct, and I can provide some feedback as to how people are doing. So if you zoom in here with plus key, you can zoom out with the minus key, you can pan over, you can see the TEM scan of the C. elegans ventral cord. So to the left we have a muscle cell, to the right we have another muscle cell, some muscle arms above, and the region which I'm circling around right now is the neural pile of the ventral cord. So if I zoom in further, you can see that the images start to get a little bit grainy, but you can work at whatever magnification level you think is appropriate. So on the left we have a group of neurons, which makes up the left fascicle, and a group of neurons that makes up the right fascicle. As you can see, the neurons have a membrane around them that stains densely, and you should be able to make them out from one another. If you have any problems identifying the individual neurons, what's going to help you out is that you can see that these are scans from prints, and so these were actually annotated by pen back in the 1980s. So if you scroll through the images with your scroll wheel, you can see how every other image will have a number on them. So if you're having trouble following, it's kind of like paint by numbers, only a little bit more technically advanced. So to begin with, what I'm going to do is I'm going to pull up the table here. So if you go show object tree, it should be blank right now, or may not have information from others using CATMAID. So what I'm going to do is I'm going to create a group underneath neuropile. And so group will pop up here. You should rename this group to identify who you are. So I'm going to call this Stephen Cook. Underneath that, you can see that there's some options. If you right click, I'm going to go to what says create neuron. And so as I said before, these neurons do have numbers identifying them. So each one of these has a specific cell identification. So for my first neuron, I'm going to click on this, and I'm going to rename my neuron, and we're going to call this neuron 31 because it's going to be the first one annotated. So with this highlighted, you can then go to the left and click here what is the tracing tool. So once you click the tracing tool, you'll see that your cursor will turn into a crosshair, and you'll be able to drop a centroid in the middle of the neuron, and that will identify the skeleton backbone for it. So making sure that you have your neuron here highlighted, again this is for organizational purposes, you can click on the image and you'll see that your skeleton 673 will show up, and that's an identifier for this object that you dropped. If you scroll through the images, you can see again, if you follow the backbone of the cell, you're able to trace through as many images. I'm going a little bit quickly here, you can go more slowly later, and you'll be able to do that. So to start a new neuron, what you can do is you can hit Control, left click, and that will stop your active skeleton that you're doing. You can create another neuron, you can call this one and rename it Neuron 42. And so you can continue with this, and you can be able to start what is now a new skeleton backbone for Neuron 42. So if you're able to do this for 2 to 5 images, that'd be great. And in my next tutorial, I'm going to show you how to identify synapses and gap junctions within the data set.